see a typical plant cell, a typical animal cell. By the way, this book is called Everything You Need to Eat Science in One Big Fat Notebook. A really good book. You should buy it. They haven't sponsored me, obviously. It's just a really good book. Anyway, this is a page on that book. They have a typical plant cell and animal cell. They're more or less the same, as you can see, but with some small differences. So, we're going to be reviewing the animal cell first. So, over here, we have the cell membrane. So, if the cell was a fort, the cell membrane is the outer wall of the fort. It has the walls so that some stuff could go in and some stuff can go out. Food, water, all that stuff goes in. Carbon dioxide, other waste products, those go out. And those are like the guards of the wall. They make some stuff go in and some stuff go out. Let's say the common cold infects this cell. How does it infect? Well, this cell membrane isn't completely flat, as you may expect. It has tiny perturbations, these tiny protrusions sticking out of itself. Now, these are typically for allowing nutrients or water to latch onto the cell and be pulled inside. But the cold virus is smart. It figured out that some of these protrusions latch on to the cold virus, so it can actually disguise as a nutrient and get pulled in. Once it's inside, enzymes, which aren't shown in this diagram but they're there, enzymes, which are just small molecules, attack the intruder and kill it. They show a small part of the virus to other cells so they know, hey, there's an intruder coming. Okay, so now that it's been infected, what's the plan of action? Well, right here is the nucleus. You can see it has two parts, the nuclear membrane and the nucleolus. So the nuclear membrane is like the cell membrane. It just covers up the nucleolus. The nucleolus is like the tank designer code for the fort. It has the codes how to design the tank. The tank is the antibody. Now many people are confused about antibodies. They do not kill the virus. An antibody just latches onto the virus and it's be like, hey, immune system, I found the virus. So yeah, you should probably kill it. So it just signals the virus is there. It does not kill the virus. Again, just clearing up some confusion there. Anyway, so we need to produce the tanks to make the antibody. Okay, so right here is a tank design code. Now, only a specific part tells how to make an antibody tank. So, we look for that part in the code and we copy the instructions onto another sheet of paper called the mRNA, messenger RNA. Now, as I mentioned in my vaccines video, the mRNA travels to the ribosomes. The ribosomes are all lined up in a structure called the endoplasmic reticulum. The ribosomes make the proteins or the antibodies. You give in the mRNA and it makes the tank. It's like the tank designer. Okay, so now we have the tank. It still needs to go out. Well, that's how it's packaged in the Golgi bodies. It's the packaging center. It covers the tank with a signal saying, hey, this is a friendly tank. It is not an enemy. You can allow it to the cell membrane. The Golgi bodies also tell the tank how to get to the cell membrane, how to escape. Now, these generate some waste process. So it's processed by the lysosome, which isn't shown here, facility of the entire fort. Now, doing all of this tank codes and tank designing and protecting the walls of the cell, this isn't easy. It needs energy. So, it starts here in the mitochondria. The mitochondria are the energy production facilities of the cell. Now, a lot of people are confused about photosynthesis and respiration, so I'll clear up one more time. You see, photosynthesis is converting light energy to glucose. Now, light energy is raw. It's like raw meat. You can't eat it. So, what the photosynthesis process does, it converts this light energy, this raw energy, to glucose. The glucose is like a container for this light energy. So this light energy needs to be refined. Respiration turns this glucose into refined energy. This refined energy is stored in a molecule called adenosine triphosphate, or ATP. And ATP runs the entire cell. So what the plant cell does, as you can see, it has these tiny chloroplasts which perform photosynthesis. They're making glucose. And the mitochondria are taking this glucose and refining it into refined ATP molecules, which can run the entire cell. But humans don't have chloroplasts. Where do we get the glucose from? From food. Food has a ton of glucose, and we use that to generate some ATP molecules. So the cell, as you can see, of a plant also has this cell wall. It gives the plant structure. It's basically like a skeleton of the plant, except the plant has no muscles, so it can't move. We have muscles, so there can't be a cell wall because that would restrict the movement of our cells. Okay? Now you might have noticed this giant thing in the plant cell. It's called a vacuole. It's basically a storage facility for food, water, and other things that the cell needs to survive. Humans and other animal cells also have vacuoles, but they're tiny, so they're basically insignificant. So we've talked about the different parts of a plant cell and an animal cell. We've talked about how cell theory came to be, and most of all, we've talked about the basic three postulates of cell theory. Okay, so let's end this episode. Dun, 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 dun. So what did we learn today? We started by learning the foundations of cell theory. We then discovered how the microscope was used to discover these foundations. We then realized that these foundations are not indivisible and actually made out of smaller parts, the sum of which is the cell, a wonderful beauty of life. So, to wrap